This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 318, recorded on August 22nd, 2024. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone. And today we have the symmetrical temperature of 82 degrees Fahrenheit and 28 degrees centigrade. And it's beautiful Chamber of Commerce weather, no humidity, and the sun is shining, and hopefully it'll stay that way for at least a couple of hours.
Get this here, it's 44% humidity. Wow, it's so low, 25 C's. We're in a little bit of a chill here in August.
Yeah.
But fear not, it's going to get hot again. Also joining us from St. Louis, Missouri, Petra Levin.
I think I'm having my own Chamber of Commerce weather. It's 77 degrees here and 46% humidity, but that will change. Next week, we're supposed to go up to 100.
Wow, 100.
Yeah, sadly. But it's great to be here in my air-conditioned office with you guys.
And joining us, TWiM adjacent from Tacoma, Washington, Mark Martin.
Hi, it's great to be here, and it is 22.3 degrees C and 60% humidity because it's drizzling a little bit.
Typical weather for Puget
I want you to
Sound.
know it's always foggy. You never see the sun. That way no one will ever come here. That's exactly what I want you all to know.
Thank you.
All right, we have two very cool papers today which involve mathematics. And that's why Mark is here. No, Mark actually suggested this first one about the species on Earth. Mark, tell us about that.
So this is a really, really interesting paper. Now, I have to tell everybody who's listening that my wife, Dr. Jennifer Quinn, is a mathematician, and she's going to be so angry at me because I can kind of talk about what's taking place in this snippet, but I think the take-home lessons are really interesting. So the name of the paper is "A Tiny Fraction of All Species Forms Most of Nature, Rarity as a Sticky State." It appeared early this year, was actually accepted electronically at the end of 2023, but it's a 2024 paper in the Proceedings of the National Academy of Sciences. The authors are Ness, Pugioni, Shetty, Stratsma, DeBow, and Levine. And the authors are at Wageningen University in the Netherlands, the Federal University.
Wageningen.
Pardon me? Wageningen?
Wageningen. And I'm probably still butchering it.
You know, Wageningen. Sorry about that. I'm an equal opportunity offender. And now I lead to the Federal University of Minas Gerais in Brazil and the University of Helsinki in Finland. And this is a very interesting paper. It is mathematical. And I got a big kick out of the authors because even though there's a lot of mathematics, they slip in humor throughout. For example, when they're describing the sticky state of what they're calling a strange attractor, more about that in a minute, they say, imagine 1,000 ants crawling on the floor and there's a sticky spot of jam. So it's not like Hotel California because the ants can leave. In PNAS.
Great. I
right.
like that, yeah.
And so what they're basically saying here is that they used a lot of data from many different sources, not just the human microbiome, but flies, rodents, fish, trees, plankton, and fungi. And it suggests over and over again that a tiny fraction of all of the species accounts for most of the biomass. So are there experiments being done in this paper? Not really. There's modeling. And it's interesting. They use this term hyperdominance. So one of the things that's so very strange about what they discuss is that you find out when you look in any population, just a few of the species in it make up most of the biomass. As you as you actually look at this, they do, for innumerate people like myself, they do a really nice abstract, which is really sweet to see. They have a significant section, which is really good. And the whole take-home lesson is this one. So they are positing that the diversity of rarer members act as insurance against changes within the environment. And I find the first sentence of the paper really sticks out in my mind. Ecologists have been puzzled by the enormous amount of rare species in nature. Now, yeah.
So Mark, Homo sapiens does not have a backup.
We do. We do.
What's
If you
that?
think about it, our backup is our appendix. It's that little bit of appendage that literally is, when you have an appendicitis, it's principally because it's chock-a-block full of stool and it's recruiting all the white blood cells to effectively burst it. And that's our effectively backup system where I think we store a lot of microbial diversity.
But backup for our species. Mark is
Oh,
arguing
yeah.
that, the rare species are sitting there waiting, like the mammals were waiting until the dinosaurs left, right?
Yes.
But who's waiting for us? Nobody, because we wiped them out. All the Neanderthals and the Denisovans and whatever else was here. Right?
I would posit that Mark's got a very nice expression for this is first evolve, last extinct.
So when you look at this paper, and I really recommend everyone do look at the paper, you'll notice that when you look at figure 1, you're seeing things that don't look terribly scary until part C. I'll come back to all this. Then part 2 seems to disturb you, and part 3 even more. But what's important to remember here is that you don't have to worry about the mathematical modeling, because they do a really good job explaining what's taking place. And again, the issue here is very simple. Why are just a few members of a particular population dominating an ecological niche? Why? And what are the rare ones? Because the idea would be some are better than others and there'd be winners and losers. And this doesn't appear to be the case. So when you go through materials and methods, they are looking at databases and they have mathematical models. And this is where, it's really bad, Vincent, because did I mention my youngest son is a senior in math and physics, applying to graduate school in mathematics this year. So now I have 2 members of my family that are ashamed of me.
Biology lost. Mark.
That is not my fault. So
the heart wants what the heart wants.
Well, yes, you're not wrong, Petra. But what I will say is that I did not try to force my kids to do biology. I had seen too much of that. And I don't want to be judgy, but some of the instruction they received in secondary school was not the way I would have done it. Wasn't that put politely?
That was very polite. As someone who has 2 children not interested in biology, that was very polite.
Anyway, what's interesting to me is that I know that my son is going to, because he hasn't seen this paper, he's going to lose it because this is the kind of thing that he does. He just came back from an REU. So one of the expressions I thought was interesting, because I'm not an ecologist. I've been called that, and it's not true. Trust me, my ecology professor at UCLA would really disagree. The idea of hyperdominance. The idea is it you have these relatively few members of a given population that kind of take over, but why are there all the rare ones there? Why aren't they excluded out? And when they do the abundance patterns, that's what you see in figure 1. And what they're interested in, if you look at figure 1, is in part A, the fraction of species needed to account for 50% of the total individuals across community as a function of the total number of species in that community. And look at that. And even I know that everything seems to be following this rule, that in any environment you're looking at, there are just a few that seem to dominate. Now, how can that be? And the whole take-home lesson comes back to something simple with all of this. The idea is, what are the other members doing? And their insurance. Their insurance against changes in the environment. And when they model it, they use this expression I like, response diversity. And they actually talk about this idea of rarity as a sticky state. And what they're really talking about is something you see in mathematical modeling called an attractor. That is to say, something you wouldn't necessarily expect, and data seems to kind of get in that area. And what I loved is I read this, and I'm looking, I believe, on the third page under rarity as a sticky state, which I wouldn't have called it. They actually say things like the eye opener comes if we add the fluctuating environment. And rather than getting a continuous distribution of states, you end up with one species being very rare most of the time. And so what that means is it's as if nature is trying to have a situation where you have a diversity of low abundance species in a given environment. And what happens is they're just insurance for some kind of a damage from the environment or a pathogen or environmental things such as we're creating all the time.
Mark, I don't think nature's trying to do anything.
Yeah.
This is an evolutionary process.
There is no
right?
intent. You're right.
But isn't it all about competition? Also, maybe the rare ones just aren't as good as the others.
And they address that. And the reason this caught my eye is I do a little work with this idea in my little lab with undergraduates. I'm interested in competition between things, and you can get fooled very easily, even very, very minor things. And you think, well, it's going to completely sweep out something that can't be there. And stochastically, you're still going to have some of those members there. And what they're suggesting is the extra rare species are not there stochastically. It's because of the way that they're dealing with small variations that take place in the environment. Because a given one is the one that's winning, for lack of a better term, is really well adapted to a specific condition. But that specific condition is not absolute.
So, Mark, with these, I'm just trying to imagine it because I agree with Vincent. Like, these are all competing with each other, right? Or, I mean, it's sort of like one dominates on a normal curve and the others are living at the edges of the normal curve here. Is that kind of the idea?
That's what they're saying.
And then, but they just can't get enough resources. But if the big one goes away, then they can take over and have a fight to the death. But there is enough for them to survive.
That's what it looks like. And they never mentioned it. What this really made me think of gasp phenomena. And so I don't know if everyone is super familiar with that growth advantage in stationary phase. And so the idea is that you have like all these mutations taking place during stationary phase, and you get wave after wave of successes over time. And it'll go in the other direction as well. So again, I say something that makes ecologists really unhappy with me, is that I don't think we understand selection very well. And that's because we can't get fine enough. We're more granular about it, I think.
Well, it's also we think about it in a stationary way. And I think that's been a big problem. That actually, this is where I'm going to put my plug in for people studying microbial evolution and microbial ecology, because you can really do the temporal stuff you really need to do to understand how this works. That's why, like the GASP phenotypes, I would say it's such a great model, because you can see evolution and adaptation happening in real time and competition.
I go back to my days working with my dear colleagues in nuclear forensics. And for them, it was all about fluence, the flow of neutrons and electrons that were coming off decaying isotopes. And it's all about energy flow. And that can influence it. And the way they modeled it is with simply Monte Carlo simulations, where everything is random, it's stochastic. And I think that's what Petra is getting at, in that the energy ebbs and flows, because unless you're just eating rat chow, the energy ebbs and flows in the natural environment are varied in space, time, quality, and quantity. And all of those are based on, who is best at competing in that particular circumstance. And I think the natural selection process has maintained this large diversity just to allow for the stochastic aspect of Mother Nature.
I think it's also that it's just it might also be a self-reinforcing system, though, because these dominant species, right, are sucking up resources and putting out waste that can then be utilized. So it's partly that nature is variable, but it's also these living systems themselves are modifying nature. So I don't know. This is just a really cool mathematical problem, but it has many variables.
So there's a wonderful thing that they put in their discussion, and they did not do this work, but they refer to it, and I want to read these papers. This is what they say. A fascinating proof of principle for the interplay of functional redundancy and response diversity comes from experimental work on bacteriophage outbreaks in cheese-making cultures. Yummy. In simple constructed communities that contain all the necessary functional groups as single strains, the outbreaks are devastating for functionality. But the diverse communities that are propagated by generations of cheesemakers, such crashes do not affect overall activity, as functionally equivalent strains compensate for such losses. So what they're saying, their view is that the remaining vast majority is important for stabilizing long-term ecosystem functioning. And the rare ones are insurance against changes. And that's not different than what you said, Petra.
No, that fits.
So, Mark, this is all done with genome
That's correct.
data, right? So give us an example. For example, in the human microbiome, what species are dominant?
In the human microbiome? There's usually the big four that you tend to see. It's not E. coli, but it's members of an, oh my goodness, you're going to correct me on this, Michael. They're no longer Bacteroides. What do you call it now?
Well, it's not the Prevotellus and the Porphyromonas. It's all in it goes up, to the next level in phylogenetics. It goes up to the order Bacteroideales.
So it's actually not species that account for most of the biome. It's something higher, right? Taxonomically.
Yeah.
Okay.
And because of the way that people look at 16S data, and since that's what we're talking about, you don't get that level of fine tuning. I do draw your attention to figure 1C here. So an example of fluctuations of dominance for a community of gut bacteria based on data from a male subject. I love this. Slightly smoothed using a Gaussian filter. That sounds like a recipe. We excluded Bacteroides that was very dominant. But if you look at this, you can see, Petra, that looks like gasp to me.
Yeah.
It really does.
But I mean, gasp is really, yeah, it does. I mean, it's not quite as pretty as the gasp model figures that I've seen, but it's basically the same thing. Like one comes up and then, yeah.
So
it does look like GASP.
As I talk,
we should put up the paper that describes GASP on the website.
Yeah, into the show notes.
Well, it's a great paper.
It's just, such lovely work. I hope to get Steve Finkel on my podcast to talk about it. And there's even a nice module for undergraduates to do the work with it. It's such a fascinating system. So the take-home lesson from all of this is very simple. When you look at any given environment, you find that most of the biomass is made up of just a few members of that community. So the question is, why do you maintain anything else but them? And the whole idea is it's kind of an insurance model. And the insurance model is suggested that when you have situations where you have variation in the environment, they act as kind of like functionally stepping in if you get most of the predominant ones knocked down. And I was so struck by how universal this was across different types of taxa. It was amazing.
The problem that I see, Mark, is that insurance is like a future thing, and evolution doesn't work that way.
No, but think a little bit.
I mean, it may be that my view would be these things are low competitors. They're just coexisting. There are enough resources for them. And then in the account of some shift, then they could take over. But I wouldn't look at them as insurance. I think it's a different view. Maybe that's a mathematician's view.
I feel like they're more backups, waiting.
Yeah.
Idling.
I mean, they can step in like mammals stepped in after dinosaurs, right? But they weren't there as insurance.
It's a really interesting idea, and I hope, as Petra has alluded, that people try to test this directly in some of the microbial systems that are available to us. I want to hear what Rich Lenski says about this myself.
I'd like to see some undergrads do this experiment in a lab. It would be really fun.
But when you're seeing, again, I mean, the real interesting thing is the dominance in biomass, which is fascinating to me, because they even say it doesn't necessarily have to do with size. There's some wonderful bit about trees in the Amazon there. It's really interesting stuff.
Mark, do you think that genome sequencing is enough is accurate enough to give ratios like this?
Every time that I have asked people about using techniques to determine abundances, I get a little bit of a soft shoe from experts, a little soft shoe dance. I think that there's a lot of, I don't want to say inaccuracies, a lot of questions about how you generate those numbers. And then there's the constant changes taking place as well. I mean, if I were to say, if I look in the human gut, exactly how many Prevotella exist in my gut, well, that all depends on how much of it was amplifiable and clonable and all the rest.
Yeah, it seems to me that you could be fooled, right? Because is it just based on 16S for say
Yes.
bacteria, or is it whole genome?
No, this is just 16S they're looking at here.
But the other species has to be whole genome because they can't do ribosomal,
right?
The 18S guy,
And
the
some of it is
eukaryotes,
just
essentially.
observational when you look at trees.
Yeah.
So I don't know. I was coming up with one of my laws.
Uh-oh.
Yeah. and my first law is that microbes have an agenda and it's not yours. And I think that that's a really important thing to remember, because I don't think that we can perceive all of the strictures that we put upon an experiment in terms of what a bacterium or a virus would experience.
Could it be something that evolution has no plan? Get over it.
Yes.
I would like to see this done with viruses, actually.
Yes,
It's an interesting point. I wonder if they're more balanced than living things.
Now, Paul Turner does a lot of excellent work at Yale involving this kind of thing. I'm curious, again, like I'm interested what Rich Lenski thinks. Paul Turner, maybe I'll write to both of them and say, what say thee?
Of course, if it's correct that in the human microbiome, a few orders or whatever dominate, then the phage that infect them are going to dominate as well, right?
Yes.
But that's a reflection of the host. So
It's specificity.
I guess I'm answering my own question. It's going to reflect the host, right? Yeah.
Yeah.
Well, I think, Petra said something interesting earlier about how organisms modify the environment and the environment modifies organisms. And that's very Eastern philosophy, but it's really true. But you know, Vincent, that viruses tend to keep one particular organism from predominating, and certainly in marine ecosystems, you get those crashes and all the rest. And many people have made the claim that they tend to promote diversity in other types of organisms just because they knock down the winners, as it were.
Yeah, I mean, everything but humans, I think that's true, right? I mean, viruses don't control our populations. We're exploding anyway, right?
Yeah, right.
But
Yet.
Yet, right.
Right. We're at a moment in time where they're not controlling our population, might be.
Actually, human population self-controls because we've run out of resources, right? So India and China population growth is slowing because it can't be sustained. I mean, the systems are just not working, not just biological ones, but, you don't have enough housing, you don't have sewage, et cetera. So, but I think in other organisms, it's probably correct. Is this testable? Is this something that's testable, Mark?
I think so. But again, it would have to be with a microbial system. And honestly, there's been so much work done with some of the RNA viruses of bacteria. Maybe that would be a good one to use. Just
Q beta. Q beta.
Yeah.
Why an RNA virus?
Well,
Exactly.
No proofreading. No proofreading.
Okay.
Yeah,
Takes that variable out.
Okay. So it might even change more quickly.
Yeah. It would. You have to eliminate the complexity so you can get at your primary question.
Right. So the idea is that it's very difficult to study evolution in real time in anything but a microbe, and that of the microbes that would change the fastest, it's an RNA virus without any editing. Just so people understand, people listening understand this very inside baseball conversation we're having.
And a lot of people have worked on bacterial RNA viruses for many, many years, so you know more about the system. And for all I know, people have done work related to this that I haven't seen.
So you Q beta people out there, this is a great problem for you.
Yeah.
It's really a worthwhile paper to look over.
Yeah, it's it's quite provocative. I think this is something I haven't thought about, and I like a new paper like that. So thank you, Mark.
You're welcome.
Yeah,
Cool.
thank you. That was really fun.
Okay. If you thought that was fun, wait till you get to this one, Michael.
Yeah, today we're doing something completely different. The title of this particular manuscript is Early Life Gut Bacterial Community Structure Predicts Disease Risk in Athletic Performance in Horses Bred for Racing. And this paper comes to us from the University of Surrey in England, and it's from the School of Veterinary Medicine, the Faculty of Health and Medical Sciences, and it's by Lang, Moeller-Levitt, Mansurge, O'Flaherty, Cook, Sells, Pinkham, Pinn, Smith, Wise, Ellis, Coutu, Alves, Laragone, and Proudman. So today on TWiM, the science we're going to discuss addresses the sport of kings, or for us layfolk, how do you pick a winning racehorse with consistency? Not only will the science of today's manuscript begin to address this issue of picking a winning racehorse, but this paper will also help all you skeptics, you know who you are, who question the worthiness of longitudinal intestinal microbiome sampling of individuals to make predictions.
That would be Elio Schechter.
That is indeed. He'll probably write a blog post taking me to task. I believe that these authors are onto something. And it's simply because they have selected outcomes that can be independently scrutinized beyond any doubt simply by asking, did the outcome result in a win, a place, or a show? After 3 years when they took the sample. So this is 3 years down the road. Did you win, place, or show? If you're not into the sport of kings, there's another outcome they use, namely overall health and well-being of the study subject. And in this particular case, it's horses, an issue that many of us non-royals fret over on a continuous basis. You know, whether your microbiome can truly impact your health and well-being. The title principally offers us the top-line conclusion of the work, namely that it's the early composition of the gut bacterial community associated with horses bred, underscore the word bred for racing, that can be used to forecast disease risk and future performance for which the animal was raised. You can almost say once you learn the fate of the microbiome of a particular foal, you can begin to anticipate that race for the roses at age 3. I probably should have saved this paper for Derby Week, but that's in May. But alas, thought that you would like to think about this today because I think it's a really great paper because it teaches us about outcomes. It teaches us about power calculations for microbiome studies. And we're all beginning this new academic year, at least for those of us in the U.S. As you might guess, this paper, we're going to discuss horses. So the first thing we have to know is the horse is a non-ruminant herbivore. These animals do not have a multi-compartmented stomach as cattle do, but they are able to consume and digest forage. The cecum and the colon, parts of the large intestine, then serve somewhat the same purpose for the horse that the rumen does for a cow or other ruminants, but with less efficiency, to the tune of approximately they're 25 less efficient than a foregut fermenter. And a foregut fermenter is where the fermentation takes place in this multi-compartmented stomach. The reason is that these are hindgut fermenters, meaning the large intestine is the main site of fermentation of these fibrous feedstuffs like grasses and hay and other things. This differs from ruminant animals like goats, cattle, deer, sheep, which are foregut fermenters with a rumen and this multi-component stomach. Now think about it. Did you ever wonder why horses don't chew their cud? Well, the esophagus of the horse has a one-way peristaltic action, which means that horses cannot regurgitate their own food and therefore can't chew their cud.
They don't vomit either, Michael?
No,
Just like mice.
just like mice. They can
Wow.
also cannot burp or pass gas
Thank
through
goodness.
their esophagus. Well, this gets back to Petra's element from earlier. It's all about fermentation balance. And where do you put your waste electrons in a cow? You put it into methane. You take CO2, you add all the electrons to it, and you belch methane. That's why they're trying to put bags over the farms in Missouri to capture all the methane. So it's all about efficiency. Now, the science of the foregut and hindgut fermentation is absolutely fascinating. Foregut fermentation is generally considered more efficient than hindgut for digesting fiber. For example, one study found that a ruminant foregut fermenter had 59% digestibility of non-digestible fiber, while hindgut only had 44%. And so the foregut fermenters then are thought to have several advantages. First and foremost, the foregutters have more microorganisms that can break down cellulose before it reaches the small intestine. And those short-chain fatty acids and amino acids that they're breaking down in the stomach actually get into the small intestine and can be absorbed by the animal. So the forage that has been partially digested in the forestomachs then enters the small intestine, so you have more efficient absorption of nutrients. It makes perfect sense when you just consider simple anatomy: stomach, small intestine, large intestine. In addition, the foregutters can convert non-proteinaceous sources, and these is code for waste products like ammonia and urea, into their amino acids. However, the hindgut fermenters like horses, tapirs, and I didn't know that the rhinoceros and an elephant was a hindgut fermenter. Did you guys know that?
Mm-mm.
So I found this absolutely fascinating.
That is not on the signs in the zoo.
No, it's not on the sign in the zoo. Now these hindgut fermenters have some advantages, including that they can consume low quality forage all day, and the size of their hindgut allows for optimal fermentation of these fibrous feedstuffs. So they can eat all day, and this large gut, large cecum and large intestine allows for the optimal fermentation of these, low nutrient materials. Now, I believe the general public is now beginning to appreciate the value of diverse gut bacterial communities. You cannot go to the grocery store anymore and not pick up a new item without stumbling into the marketing term prebiotic and probiotic, prominently displayed on their labeling. Well, that's because there's a growing body of evidence to support the critical role of early life gut microbial communities in the development of immunological competence, susceptibility to our disease processes. And if you think about it, it's why everybody is now appreciating the value proposition that breastfeeding offers our infants. It's why it's so important, because we're providing the human design food to the new humans. So much of what we know is because of the advances in inexpensive sequencing that has enabled the curious to test and then develop a better understanding of the role of gut communities play on their hosts. Of course, the literature is filled with numerous studies mapping fecal bacterial communities to different compartments of the equine hindgut, the so-called hindgut fermenters, and recall that they're 25% less efficient than the foregut fermenters. And importantly, just like in us humans, there's evidence that characterized changes in the microbiome has been associated with intestinal disease. Now, the authors offer that early life colonization of the full gut, so this is the baby horse, has been described, but the influence of the microbial community on the health of that animal later in life and performance, whether it could win a race, is unknown. Since I'm not of royal lineage, no one's ever called me Prince Michael, I had to look this up. A foal, Mark's laughing. A foal is either a horse of either sex less than a year old. A yearling is a horse of either sex that is between 1 and 2 years old. And a colt is a male horse under the age of 4. And a filly is a female horse under the age of 4. Now, the authors offer that the racehorse industry is the second largest rural industry in the United Kingdom after agriculture, as assessed by economic value as measured by employment. And it has a revenue stream of approximately 4 billion pounds or 5.2 billion US dollars. So any understanding of what might minimize the risk of disease and injury in this industry is important to help them understand the welfare of horses used for racing. And then probably the take home is maximizing the potential for athletic performance is key to successful racehorse breeding and training businesses. Second, the relationship between gut bacterial communities and athletic performance is unclear. However, studies in mice, rats, dogs, and even horses have already produced evidence that changes to the gut bacterial community can result in a response to exercise. There's less evidence that the gut bacterial community structure is associated performance, and that's what they're going to embark on. So, as you might surmise, the authors developed here, they looked at the fecal bacterial community structure from a large cohort of foals bred for racing, along with the comprehensive health and performance data of the sample foals for their first 3 years of life to determine associations between early life gut bacterial community structure and future health and athletic performance. Now, this paper's got a really great method section. They take you through how they did this. So 57 thoroughbred foals bred for flat racing, born between the beginning of February and the end of May, were recruited from 5 thoroughbred stud farms in the UK. They then used an a priori sample size estimator for paired differences in means, calculating the R power package that indicated that they would only need to look at the stool or manure from 36 foals, and that would provide more than an 80% power to answer the question to a medium effect size with a 95% confidence level. But because they were concerned about incomplete sampling, would they get all the data, and losses to follow-up over a 3-year study period, the authors elected to over-recruit the number of horses in their study by at least 50%. So there's a total of 57 foals that come into the study to begin with. Typically, the foals were weaned at about 5 months and then spent their first year on the stud farm of origin. All of this is important because it introduces the variability and the selection pressure. Between 9 months and 1 year of age, the yearlings were typically moved to other premises prior to their commencement for training for racing at around 2 years of age. And so they ended up at 29 different training yards with no more than 3 horses at the same location. Again, why is this important? The simple answer you may recall from our TWiMs on wine, namely it's terroir. It's the French term that describes the unique environmental factors shaping a wine's character. And it's important that we got 29 different sites that they're grazing on, and it includes soil composition, which will influence the forage they're eating, climate, microclimate. And here, of course, with the horses, it's what they're grazing on and other supplements to their diet. And since there's 29 different training yards, they're all probably doing something different. So then they get written and verbal health updates were obtained on a weekly basis for all horses, reporting any veterinarian investigation and or treatment for orthopedic injuries, soft tissue injuries, respiratory illness, or gastrointestinal
They're better
disease.
cared for than humans.
Yes. Well, I was going to get to this, but the average foal costs about 30,000 pounds.
Yeah.
That's not for, secretariat style horse flesh. This is,
Average.
your average, So what they did, the health updates were obtained. Again, just like a human trial. Observations were censored for all study participants at 156 weeks. And the registered racing names were used to track race performance using a public access database, racingpost.com. And I dropped that into the show notes. And the outcomes that they looked at were finishing position, official rating. Now, you can't confuse the figures. If you're one of those people that only looks at figures first, OR doesn't stand for odds ratios here. It stands for the official racing rating, which is a handicap metric determined subjectively by an official of the regulatory authority of the UK that rates increases are related to the quality of the horse's performance. This was obtained after every race start. They also measure total prize money earnings and cumulative performance metrics as assessed by how many times the horse start a race, the total placings, whether they were first, second, or third, win, place, or show, and the total number of wins. So they collected all of those until December of 2021, which was the end of the season. So the other methods that they use are well familiar to routine listeners of TWiM. They use 16S ribosomal RNA targeted metagenomic sequencing, or NGS, using the Illumina MiSeq platform. But in contrast to what we use in the infectious disease world, where we use V1 to V2, here, because we're looking in the large intestine, they used V4 and V5 simply because that primer set is better at characterizing microorganisms that harbor that particular niche. Now, their microbiological results. Bacterial diversity, as measured by alpha diversity, was low at early sample points. That makes perfect sense. They're feeding off of horse milk. And this increased until 60 days, which was sampling point number 5. The significant differences in the two metrics were bacterial community diversity, observed bacterial features, and Shannon Diversity Index. Since I anticipate that this episode may have a provocative title like, how do you pick a winning racehorse? So we may receive more lay listens. Shannon diversity is most easily imagined when comparing a composition of a forest to that of the microbial community we're characterizing. This index measures two things: richness, which is the number of different types of trees in the forest, and then evenness, how equally distributed the trees are. For example, if it's mostly pines or a mix of oaks and maples and pines. And so Shannon diversity is an easy way to measure both richness and evenness in a microbial community, much like the forest sample. So a higher Shannon diversity index means there's a wider variety of microbes and they're more evenly distributed within the community, where there's a large body of literature offering that individuals with a high Shannon diversity, and this is both for horses, people, everything that we've looked at, are often healthier. So a higher Shannon diversity suggests that the animal is healthier. A lower Shannon diversity index means there's fewer types of microbes or that one even dominates. And this can be a sign of an imbalance, which might lead to health problems, which ties back nicely into Mark's paper from earlier. So this is what often happens as well in the development of periodontal disease. Oftentimes, we'll see in a periodontal patient, they get this bloom of these really bad gram negatives over all of the other good microbes that you need in your oral cavity. So the Shannon Diversity Index is simply this easy tool used to understand how complex and then balanced any particular microbial of an individual, this is an individual measure, is. So take us to panel A of their first figure. And it goes from sample point 1, which is, effectively the newborn foal, all the way out to sampling point number 9, which is at 365 days. The sweet spot is sampling point number 4 at 28 days. And when you begin to look at this figure, and it's just an easy heat map, you don't have to know much about interpreting heat maps other than the fact that the more colors it suggests, the greater the variability. And you can see that that color change really begins to manifest itself at about 28 days, where the animal is beginning to do some milk as well as some forage, and the microbial community is changing. Now, the families were principally Enterobacteriaceae and Bacteroideaceae were the most abundant at 2 days old, and that's effectively the microbes that eat milk. The bacteria from the orders, now we're going, remember, family is below order. It goes kingdom, phylum, class, order, family, and genus and species. So if you keep that straight, bacteria from the orders Bacteroideales and Clostridioides were the most abundant in samples from a later time, which is suggesting that those microbes are effectively processing forage. So many bacterial families, including Spirochaetaceae and Fibrobacteriaceae, were not detected at early time points. Again, makes perfect sense if you know what they're eating, but increased in abundance as the foals age. Now, their top-line conclusion from this microbial analysis, when they integrated the data with performance, diversity predicts not only the risk of disease, where more diverse means less disease, and then using a survival analysis as their metric, they evaluated the effect of fecal diversity had on this rate, and they looked at the first episode of respiratory disease, something that's easily measured. Simply put, they're using correlative observation for respiratory, soft tissue, and orthopedic health events, and they were most often associated with an incident rate of disease where it was found that they had lower gut bacterial diversity in early life. And they got a really easy figure to look at. I mean, it's like a stoplight, except they're not using red, amber, and green. They're using shades of blue, which I appreciated being colorblind.
I think this correlative data is really worth thinking about for a second, because
Yes.
there's a lot of correlative data with microbiome and health. And at this point, it's just correlative, which is sort of like saying, it's like AI, right? It looks for patterns, and this is a pattern. But does this pattern mean something that we can get more information from or not?
And that's what goes into the next section of the paper that are going to get people who own a lot of racehorses really excited, namely that diversity predicts athletic performance. How, you might ask? Simply, the data they have collected offer a unique opportunity to simply ask the question as to whether the early life gut microbiota diversity has an effect on subsequent athletic performance. And again, Petra's absolutely right. This is all correlative. Here they used a general linearized mixed models where they're fitting them using three-dimensional outcome variables, which are proxies for athletic performance. They were also using the official rating or OR from the race handicappers, and they were using average prize money earnings. Now, the thing that they're going to find is day 28, the stool that you see at day 28 is going to tell you how well that horse will likely do when it's 3. That's pretty good correlative data. So here they found significant positive associations between the fecal microbial diversity from the day 28 time point, or SP4, was associated with all 3 outcome variables, with some measures of diversity at 90 days being also significantly associated with performance. And that's in their figure 3, figure 3A. In panels figure 3B and D, you can see whether you want to place your own funds at risk based on this diversity index. When you look at the regression lines and you have to pick on the right side or left side, of the 95% confidence interval, I can almost imagine Mark putting that as a quiz for one of his students. Which horse would you best bet on based on this graph? Mark's giving me a look. So
I'm just saying.
I'm
just saying, why the long face?
Oh, yeah. The last component of their manuscript concerns the use of antibiotics and its effects. These are expensive animals, as I said earlier. Training costs $30,000 to £42,000, so any infection in these young animals could be devastating to your investment. So they looked at how many of the horses were exposed to early life antimicrobials. This is in their 4th figure. Here, the top line conclusion was that foals receiving antimicrobial treatment during the first month of life had, again, no surprise here, significantly lower fecal bacterial diversity at day 28. But what was very interesting was that in their subsequent racing careers at 3 years, the average prize money earnings, a proxy again for athletic performance, was significantly lower in foals subjected to exposure. And the distribution of official ratings by the racing authority differed according to antimicrobial exposure. So I said to myself, shut the barn door. Needless to say, I was not surprised by their findings, but was impressed to see this correlation held all the way out to 3 years. But unfortunately, it was not statistically significant. But given it was only based on an initial sample size of 57, and all of those individuals didn't receive antibiotics, I wonder if it warrants a bigger sample size or a different style experiments.
Michael, it seems it would be useful to do this going forward for years, right? Assuming you can get, it's just feces, right? So it's not hard to collect and then correlate that with health and racing results for many more animals for a longer period. I think that would be interesting because I worry of confounding effects with such a small number, right?
So one of the projects I had as an undergraduate is I worked on cave spiders, and we grew the spiders on horse manure. And there was a difference between choice and prime manure. Prime manure, the graduate student or the undergraduate had to go behind the horse and catch it before it hit the ground. That's prime stool. Choice was where it hit the ground and it was still steaming. And good was it hadn't dried out, but it was on the ground.
So if you have to stand behind and catch it, you have to be careful you don't get kicked.
Oh, yes.
Yeah.
Oh, yes.
And Vincent,
yes.
Vincent, the jokes write themselves.
So the final data they offered was in response to the findings that the microbial structure had a role to play with respect to later in life health performance. Here it was a simple comparison, and I really like this. Again, they used the Shannon diversity measure, and foals were categorized as either having a high Shannon diversity, middle, or low, and they based it on the 28-day criterion sample. And the three groups of horses had significantly different Shannon diversity indices, and they were able to effectively find that low and middle diversity samples were dominated by bacteria from the Ruminococci on average 21% of the total reads, although this did vary between individuals. Many of the samples with the low diversity samples comprised over 40% of a single bacterial family. And now here they use non-metric multidimensional scaling, which is a really fancy statistical modeling program that there's an analogy for it, but for the sake of time, I'm not going to go into it. But you can Google it and it'll come up. So you might ask, are the funders of Royal Horses thinking about developing a probiotic feed or winning feed for an animal? Well, of course, that's another study, but they did the next best thing. They asked, based on generalized mixed models, with particular bacterial family abundance as the exploratory variable using the day 28 time point, the higher relative abundance of Anaeroplasma taceae were associated with higher official ratings in the racing form, and a higher relative abundance of Bacilliaceae were associated with higher race placings. So granted, they were only looking at this in a family sense. They didn't do any experiments here where they fed a probiotic of Anaeroplasmaceae. But I'm sure that a royal household is out there who's likely funding the next study where they're likely to index this to perhaps the genus species level. So in summary, how do you pick a winning racehorse? The answer is you collect the manure on day 28, you develop the Shannon diversity index number, and you ask who's there, and that this community structure is a critical window. Day 28 seems to be the magic number. And so they're modeling data. The authors authored some of the evidence associated with between diversity at 14 to 16 days can impact other health outcomes like orthopedic and soft tissue and GI complications, although the signal was a little bit less consistent. And finally, they offer the limitations. And again, it goes back to what we were talking about with Mark's paper. This is only 16 level, albeit metagenomic, targeted metagenomic sequencing. So the granularity of the health event data and the imprecise nature of measures of racehorse athletic performance. But still, it was a very cool paper to read and to think about all the models. I'll end by quoting the last line from the author's discussion. For the first time, this study demonstrates a relationship between early life, gut bacterial communities, and subsequent athletic performance has implication for all athletes for species, including humans, end quote. So you can almost see all the tiger moms and dads thinking about training their child to be that next gold medal sprinter or even a mathlete.
I think, the people who bet on racehorses, they get a book with
Yes.
all the information about the horse, right? And this is going to be added to it, the Shannon index.
Yes.
And they're going to be like, what is this? Can you tell me what this means?
They'll probably be better at explaining it than us.
I don't know.
But this idea of probiotics, I wonder if that would be ethical because, this is highly regulated. You can't give your horse stuff. And so I don't know.
But I mean, a lot of probiotics are pretty simple molecules. I don't think that it's, I don't know.
One
That's
project
a good
I
question.
had in the mid-90s, we were characterizing forage to increase the body weight of animals. And we learned how to do that. And without having to do feeding trials. I can't talk about what we did because it's behind the secret wall, but you can figure it out.
But I feel like a lot of this comes down to a healthy microbiome helps you be a healthy horse or person or whatever. And we're still left with why. And what is it that the microbiome is doing? Is it nutrient? I mean, my guess is it's not the microbes, it's how they help you utilize your food.
Yeah,
so, I mean, I think the next step might just be the horses actually would be a good place to do this because they don't have as varied a diet as we do, as far as I know.
Or as complex of a digestive system.
Exactly.
The fact that they don't have these multi-chambers that are effectively doing sequential fermentations, if you will.
Right. So, I mean, are they better able to extract the protein and other necessary nutrients? This is all interesting, but in the end, it's sort of like how many studies can we have, right, that tell us, good Shannon diversity is good. A microbiome with good Shannon diversity is better than one without. But the question still remains at the mechanistic level.
Mechanism, yeah. They say that in the discussion. I have no idea.
Absolutely. Yeah. It's cool.
So, Mark, do you have day 28 of your son's diapers?
No.
To know the mathlete versus the musician?
No. And
that's good.
I kept waiting for you to make jokes about fecal transplants and all the rest. It didn't happen. But
I was good.
Yeah, no, you weren't. But it's okay. I think you bit off more than you can chew. There you go. We've got lots of that. We'll get you in a harness for another one. How's that?
Thank you, Mark.
You're quite welcome. No, I mean, these are wonderful studies that people do. What I like here is that sometimes they're just observational. I mean, a few years ago, well, I guess it's more than a few, there was a wonderful skin microbiome study. I'm not making this up, from roller derby participants, where they're looking about the changes that take place, presumably from, hitting one another. Now that's different than this. What you're doing is you're looking at what's, you have winning horses, you're studying different things about them. Actually, I don't know, this idea of feeding them, it's such a regulated industry. There's been so much opportunity for corruption over the years, so many incidents of it as far back as you go to. And I'm not basing that on Hollywood. It's there's a lot of money changing hands. So my theory is if they decided there was a probiotic that worked well, that would probably be highly regulated.
Mark, in light of your paper, it must be that the minority members of these microbiomes are making the difference, right?
I think that's a really interesting way to ask and ask, whether or not there are particular minority components that are associated with things. Yeah. Wouldn't that be horrifying if that's what you have to look at?
Yeah.
Well, I mean, I don't think it's horrifying. It's harder, but
yeah,
it would make sense because if all the horses share a few members, right, or orders, then it has to be the minority ones that make a faster horse or a healthier horse over the others.
Or is it just the diversity in general so that you cover? Mm-hmm. It might just be the diversity, because I doubt that maybe it is like one species, but I feel like it's probably more the diversity of metabolic activity.
So Michael, you're quite a horse fancier, it sounds like to me. No joke is coming your way. I am curious about whether or not horses do coprophagy.
I don't know if they do or not. Mice do. Mice, I know, do. Yeah. I don't know if horses do. Dogs do.
A lot of ruminants do. And so that's something I don't know. It had to have been something they thought about, because if they're looking at fecal samples and you have other animals consuming that, right, then that would be transferring over. So it takes you back to the earlier stuff. I mean,
we don't know the other horses that are on the farm because they send them to 29 different farms. So there's a lot of really neat questions that come out of this that you could well imagine asking the investigators during a seminar, and the discussion would be really quite rich, I think.
So Dr. Google says in answer to your coprophagy question that foals and young horses do, but that it's a sign of maybe a problem or deficiency in older horses. So maybe the foals have the right idea. They're trying to be as fast as possible.
I was just going to say that my guess is there would be ample funding for this project.
Mm-hmm.
Yes. Mm-hmm. I wonder if there's a similar one out of Kentucky.
I'm sure.
But also there are other animals that race, right? They could do it with them, dogs, right?
Yeah.
Yeah. I like them doing it with horses. It feels better to me.
Yeah, but it's just well, I don't like dog racing at all. I think that's weird, but it's just a fecal sample. It's not invasive, so it's not a
Yeah.
problem. But you could do it with any kind of performance-based animal
I
or
wonder if
human,
we should
whatever.
do it with athletes,
Could do it with
You
athletes.
could.
like
Yeah.
distance endurance runners. Although the
Yeah.
horses, I guess, are more in a sprinter.
Horses
Well,
are sprinters.
they're kind of like
yeah.
mid-distance.
I think
They don't have the humans can beat horses in long races.
yeah, but I think
Yeah, but
Petra, I would love to see that. I think that'd be very interesting because there are certain groups that are really, really good at distance running, and I'd be curious if there are differences or similarities.
yeah, I would be curious. I also would be curious for the sprinters too, because again, like the horse distances are closer to like a 400, 800 meter runner. But I also would be curious because I'm sure this data is out there. People have done microbiomes of athletes. There was, I think, from George Church's lab, this idea that some spin-off project that they were going to get the microbiome and then you could eat it. You could do your own fecal transplant. But I mean, if it is diversity, that's much easier to imagine how that works and how to mitigate that than if it was specific species.
Yep.
So,
Right. Thank you, Michael.
Hey, you're welcome.
That's TWiM 318, show notes, microbe.tv/TWiM. Questions, comments, TWiM@microbe.tv. If you like these discussions here on TWiM and all the other podcasts at Microbe TV, we'd love to have your support. Microbe TV is a 501c3 nonprofit, which means that in the United States, your donations would be federal U.S. tax deductible. Mark Martin is at Matters Microbial, which is a Microbe TV podcast, and the University of Puget Sound. Thank you, Mark.
You're welcome.
What number are you at now? 53?
We just recorded 55.
Very good. I'm looking to 1,000. You do one a week. It's not forever.
Yeah, I'm doing one a week.
All right. Michael Schmitz at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levins at Washington University. Thanks, Petra.
thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.